All right, today we are diving deep into what is truly one of the most remarkable processes in all of nature, the very first few weeks of human life. We're going to unpack the intricate molecular dialogue, cellular invasion, and the precise genetic blueprint that turns a tiny cluster of cells into a developing human. Let's get right into it.
So here's our roadmap. We'll kick things off by looking at how the uterus gets ready for its arrival. Then we'll walk through the mechanics of implantation itself. After that, we'll see how that all-important lifeline, the placenta, gets built. From there, we'll zero in on the embryo's own lightning-fast development. And to wrap it all up, we'll connect this foundational science to what it all means in a clinical setting.
You know, everything really starts with a conversation. Long before there's any physical contact, there's this complex molecular back and forth happening between our two main players, the tiny, free-floating blastocyst, and the receptive lining of the uterus, which we call the endometrium.
Now, this conversation can only happen during a very specific, very brief period of time, the implantation window. We're talking about a short 5-day stretch when the endometrium is perfectly prepared to receive an embryo. Timing here is absolutely critical. If that blastocyst shows up too early, or even a little too late, that window is closed and a pregnancy just can't move forward.
So what opens this window? Well, it's this hormonal symphony, conducted mainly by estrogen and progesterone. Estrogen goes first, preparing the surface by thinning out the protective mucus and basically waking up the cells to be receptive. Then, right after ovulation, progesterone takes the stage. It cues the growth of these tiny cellular fingers, called penipodes, that help the embryo dock, and it enriches the tissue underneath with blood vessels, creating this perfect, nourishing bed for its arrival.
Meanwhile, the embryo has its own prep work to do. See, the blastocyst has this outer layer, the trophectoderm, and then the inner cell mass, which is what will actually become the embryo. But the whole thing is trapped inside a protective shell called the zona pellucida. To have any chance of implanting, it has to hatch. So the trophectoderm releases enzymes that basically digest a little escape hatch in the shell, letting the blastocyst squeeze out, ready to make its move.
Okay, so both sides are prepped, the blastocyst has hatched, now the main event can begin. The best way to think about the physical process of implantation is like a carefully staged 3-act invasion, where the embryo systematically breaches the uterine wall.
Act 1 is apposition. This is just the initial loose docking. The blastocyst just kind of settles onto the uterine wall, often guided in by those penipodes we just mentioned. Act 2 is adhesion. Here, that gentle connection becomes a firm, sticky bond, thanks to a whole family of adhesion molecules called integrins. And for the finale, Act 3, invasion. This is exactly what it sounds like. The embryo's outer cells actively push their way between the mother cells, literally burrowing into the uterine lining to embed themselves.
And what's really amazing is how this invasion starts. The embryo's outer trophoblast cells, they actually start to fuse together, forming this highly invasive multinucleated tissue called the syncytial trophoblast. Because it has no cell boundaries, it can act almost like a single giant amoeba, extending these processes that digest the maternal tissue and pull the embryo deeper and deeper inside. It's incredible.
And this whole dance is choreographed by a complex molecular language. The embryo pumps out HCG, which is the famous pregnancy hormone. It doesn't just tell the mother's body it's there, it also preps the endometrium locally. You've got integrins acting like molecular Velcro, growth factors that boost receptivity. And then there's MuC1, a protective mucus that the embryo actually has to clear out of the way before it can make that direct cell-to-cell contact needed for adhesion.
All right, once the embryo is embedded, the whole mission changes. It's not just about getting in anymore. Now, it's about building a permanent, functional lifeline to the mother's blood supply. This is the very beginning of the placenta.
Those outer trophoblast cells now take on 2 totally different jobs. Some become the villus trophoblast. Their job is to build the chorionic villi, these intricate, tree-like structures that are the sites of nutrient exchange. These guys are basically invisible to the mother's immune system. But then you have the extravillus trophoblast, or EVTs. These are the real invaders. They migrate deep into the uterine wall, anchoring the placenta, and this is key, remodeling the mother's arteries to create a high-flow, low-pressure pipeline of blood for the baby.
This picture really shows that process beautifully. First, you see these little chambers called lacuna form in the invading tissue, and they immediately start to fill with maternal blood. Then, columns of other trophoblast cells push out into these blood-filled spaces, forming the first primitive villi.
Over time, these structures will just keep branching and growing, eventually getting filled with fetal blood vessels, creating that huge surface area the placenta needs to do its job.
But the mother's body is not just a passive bystander here. In response to this invasion, the uterine lining undergoes a massive transformation called decidualization. The cells become plump, they start secreting nutrients, and they form this dense protective matrix. This new tissue, the decidua, plays a really clever dual role. It nourishes the embryo, but it also contains it, acting as a barrier to make sure the invasion doesn't go too far and harm the mother.
Now let's clear up a common myth. We find a ton of immune cells here, specifically uterine natural killer cells, or UNK cells. That name? It's a bit of a problem, because it's totally misleading. These are not the same killer cells that you find in your bloodstream. In the uterus, they have a really weak killer function. So the myth is that they're there to attack the embryo. The reality? They're essential regulators. They actually cooperate with those invading trophoblast cells to help remodel the maternal arteries properly. They're builders, not killers.
So while this lifeline is being built on the outside, our focus shifts inward to that inner cell mass. And what happens next is just this explosive period of organization, turning a simple clump of cells into the recognizable blueprint of a human body.
I mean, just look at this timeline. It is absolutely breathtaking. In just a couple of weeks, the whole thing transforms. Around day 13, it's just a simple 2-layer disc. A few days later, it folds into 3 layers and immediately starts building the nervous system. And get this, by day 23, a tiny primitive heart is already beating. By day 35, the embryo is only 7 to 9 millimeters long, but all the major body systems have already started to form. It's just staggering.
The most important event of that 3rd week is something called gastrulation. This is where the embryo goes from being a flat, 2-layered disc to something much more complex. Cells from the top layer migrate inwards, creating this groove called the primitive streak. This movement establishes the 3 primary germ layers, the ectoderm, mesoderm, and endoderm, which are the founding tissues for every single part of the body. It's a profound moment that sets up the entire head-to-tail body plan.
And then in week 4, the big story is neurulation. That flat sheet of ectoderm on the embryo's back thickens up, folds in on itself, and then zips up to create the neural tube. This is the very beginning of the brain and spinal cord. And as this is happening, the whole embryo, which used to be a flat disc, starts to curl into that classic C shape. And you can see the first tiny little buds where the arms and legs will eventually grow.
Now, all of this incredible high-precision construction is directed by a genetic toolkit. You have homobox genes, which are like the master architects, telling each segment of the embryo what it's supposed to become. Wnt genes are like the project managers, handling all the cell-to-cell communication. And then there's the famously named hedgehog gene family. Yes, including one called sonic hedgehog, which are critical for patterning the brain and spinal cord, and even telling the body it's left from its right.
Okay, so let's pull all of this amazing science into the real world. How do we actually monitor these delicate early stages? And what does it look like when these incredibly precise processes, well, go wrong?
This right here is what a clinician is looking for. At 6 weeks of gestation, which is about 35 days after ovulation, that developing embryo we've been talking about finally becomes visible on an ultrasound. We call it a fetal pole. Seeing this is the first visual proof of everything we've just discussed, and often at this stage, you can even detect the flicker of that tiny S-shaped heart, which confirms that you have a viable pregnancy inside the uterus.
But as you can imagine, these early weeks are incredibly fragile. An ectopic pregnancy is what happens when implantation occurs in the wrong place, usually the fallopian tube. A blighted ovum is when the placenta starts to form, but that inner cell mass, the embryo itself, never develops. And a biochemical pregnancy is a loss that happens so early it's only detectable by hormone levels before anything could even be seen on an ultrasound. Each of these is a failure at a very specific point in the journey we've just laid out.
And this slide really brings it all home, showing what happens when all these processes go right. On the left, there's that fetal pole at 6 weeks. On the right, just 7 short weeks later, you have a recognizable fetus with a head, a body, arms, and legs. This absolutely incredible transformation is the direct result of that whole complex journey of implantation, placentation, and early development we've just explored.
So I want to leave you with one final, kind of mind-blowing thought. We now know that for nearly the first 2 weeks, the human embryo is essentially on its own. It directs its own development, forming its first layers and structures autonomously without any direct commands from maternal tissue. It just knows what to do. Which leaves us with this incredible question. How does a simple ball of cells already contain all the information it needs to self-organize to kickstart what is arguably the most complex construction project in the known universe?